The following is a conversation with Professor Simon Carding, Group Leader at the Quadram Institute and Professor of Mucosal Immunology at the University of East Anglia. This is Inside Matters. My name is Dr. James McIlory. I hope you enjoy it. So, how did you get into the field of the microbiome?
So by training, I was a mucosal immunologist. So I was interested in many years looking at the properties of esoteric populations of immune cells in the gut. And then I came across...
Sorry, for the listener, what is a mucosal immunologist?
So a mucosal immunologist is somebody who's interested in the immune system in our mouth, gut, reproductive tracts at the boundary layer of our bodies. So the first line of our immune defense is at mucosal surfaces, and the populations of immune cells that live in these tissues in the gut reproductive tract are quite different from those you find in the blood. So their particular design is to combat and deal with infections before they spread through the body. So they have different populations and different functions. And in fact, the biggest population of immune cells in our body is in the gut. It's a huge immune organ.
Wow. Is there like a percentage we could put in it, or?
It's, I think, 60 to 70% of our immune system is in the gut.
Unbelievable.
Yep. And then the lungs have its own unique population of immune cells, the reproductive tract does. But interestingly, all these different sites communicate with each other because the immune cells circulate from one to the other to try and give us global protection at all these body surfaces and entry points.
And if there's been an invader in one part, do all the immune cells go, uh-oh, something not right there, and can they migrate and sort of move around, or is it localized to the specific area?
No, I mean, the whole purpose of the immune system in these organs is to contain and eradicate. So if there's an alarm bell ringing in the gut and there's potential exposure to the same thing in the lungs, you will get cells moving from the gut to the lungs to provide protection at those sites as well. So it's a highly intricate and highly mobile response unit, if you like.
Mm-hmm. And we'll go back to your career progression in just a second, but there's microorganisms all over and inside your body, right?
Yeah.
So how does the immune system, or the mucosal immune system, recognize that they're not baddies?
Yeah, so this is what immunologists would call it immune tolerance. Microbiologists say it's microbial tolerance. They're two sides of the same coin. And so we don't really understand it, but what's interesting, if you look at the lifespan of an individual, we are born sterile, so there's no way of educating in the womb there in terms of what's good and what's bad. So once you're born, your body gets very rapidly populated with microbes. Hopefully, they're beneficial microbes you acquire from your mother. And as those microbes start to populate all the body surfaces of the infant, the immune system kicks into life. It starts to mature and develop. And it's developing in parallel with your microbiome. And what we believe is happening is there's an education process going on there. So the immune system learns to differentiate and react or not react to the microbes that are colonizing the body. And by about 3 or 4 years of age, the immune system is at a state where it can now recognize the microbes that inhabit the body, the microbiome, as being harmless and beneficial, so it won't react to them. And yet, if there's an invader coming in, it has now acquired the ability to respond to it.
Right.
So we believe a lot of this education process is occurring within the first 4 to 5 years of life.
Fascinating. So those early life years are just...
It is. And
critical.
So, of course, what we need to do is protect that window of development. And, of course, what we know is a lot of children of that age get exposed to lots of antibiotics, which can either set back this process or completely throw it off kilter.
Wow.
And we believe that the chronic exposure to antibiotics is disturbing all this careful balance, and that can predispose to diseases later in life. And the sort of example of the paradigm for that is atopic disease, allergies, maybe even asthma.
And that relates back to the hygiene hypothesis.
Hygiene hypothesis, yes. Yeah. So, whenever I give public talks about the microbiome, and I always say at the end, let your children play in the dirt, eat dirt, get as much exposure as possible to diverse microbes from pets, let them have pets, let them play in the garden, these things. It all helps establish a healthy microbiome.
We had someone on the podcast talking, it was Debbie Shawcross, talking about how we were too clean during COVID. And her concern was that there was this tidal wave of atopy, can I call it that? Atopic disease that's coming. Because that cleanliness obsession had an impact on our kids during that really, what you're describing as just a critical period of their lives.
Yeah,
right?
And that's the problem with these sterilizing agents, the hand gels.
Yeah.
And even things that antibactericidals that go into our soap powders and soaps, they are having a detrimental effect on your normal healthy skin microbiota. And so what it does, it wipes them out and leaves spaces for pathogens, viruses in particular, to invade, because that barrier you've just wiped out with a sterile solution. So, it's good in one way, but excessive use of these things is in no way a good thing.
This is probably quite a controversial question, but should we even be using soap at all?
So, when I was growing up, it was carbolic soap, right? And that seemed to work quite well. But now we have antibactericidal soaps and things, which I think has just gone too far. We've taken the hygiene too far, and we're compromising our microbiomes, and particularly giving us the protection to infection that we would otherwise have.
And what about cleaning our food and pesticides that we use and that kind of thing?
Yeah, so there are some interesting links to pesticides and toxins in food which are disturbing or interacting with the microbiome. But it's a careful balance, because you obviously want to protect the safety of your food. You don't want to be exposing them to nasty pathogens like Salmonella, for example, or Campylobacter. But on the other hand, there are potentially beneficial microbes that are present in the soil that get into the food, which can have a beneficial effect on your microbiome. So it's a compromise. It's like in everything, a little bit is good, but too much is potentially bad.
And I guess people might be listening to this going, okay, I need to run around and roll around in the soil. But even in a teaspoon of soil, there's as many bacteria as there are stars in our galaxy. So maybe it's just about not going too far in the other direction, but not being afraid of a bit of dirt.
Yeah, and I think it's sort of, we're talking about a lifelong, continuous, low level of exposure. It's perfectly fine. Drastic change in your lifestyle, like immersing yourself in mud baths and things, that's too much too quick, and that can have a bad effect. It's just overwhelming. But if you're used to gardening, for example, if you're doing a lot of gardening, then you're getting a lot of exposure to it naturally. Yeah, and that hopefully is sort of persisting throughout your life. You're continually exposed to that outdoor environment.
I'm just wondering, is there less atopy in gardeners? Do we know?
Well, there is in children that are raised and born on farms. Than those that live in sort of built urban environments. Wow. And we relate that to sort of hygiene and cleanliness. And what are we not getting exposed to living in, a city that you would get exposed to in, the countryside, for example.
But would there not be maybe more bugs in the London Underground than on the farm?
Some quite nasty ones, I would think.
Probably the nasties, right? Yeah.
No, it was interesting because a survey done a few years ago of New York subway, where they went around taking swabs and things. Goodness. And a lot of them were from fecal contaminants, but also they identified completely unknown potential pathogenic organisms living on surfaces in the New York Underground.
So if you're going to wash your hands, you should do it after the New York Underground. Yeah. Or the London Underground.
Oh, I've been on the tube.
I feel just dirtier going on the tube. And people probably relate to this. You just feel a bit...
Yeah, in the line of work we do, you can end up being paranoia. You think, I know so much about what's out there and the potential of it impacting on my health. I don't want to go out. Yeah, I'm going to continually wash my hands. The Howard Hughes syndrome, if you like. You isolate yourself in a bubble and you don't allow people in direct contact because you're so worried about the potential risk of getting exposed to a nasty organism.
But that's wrong, because you've got your immune system.
That's right.
You're probably going to be fine.
And once you start to sort of reduce the exposure, your immune system will naturally get to these pathogens. It's like a muscle. You've got to keep exercising and working it to work effectively. Once you stop, that's when your immune system can potentially become compromised and leave you open to infections.
It's the first I'd heard that analogy before, but I really like it. You've got to train it.
Absolutely, you've got to train it.
So actually, let's go back to your journey before I go off on a big tangent about training your immune system muscle. But we need to talk about that because I love that analogy. So you trained as a mucosal immunologist.
Yep, I did many years ago. And I was working on these very strange populations of immune cells. But then I had the great fortune to come across an individual called John Sebra, who was at the University of Pennsylvania when I was there. And we met at an internal meeting, and we got talking about my interest, which was gut immunology, his interest, gut microbes. And he said, we should come over and start talking. And on one visit, when I went to his lab, he said, I've got to show you what's in the basement. Yeah, what's in the basement? And in the basement, he'd built himself a germ-free animal isolator out of bits of plastic tubing he got from B&Q, home base. The thing worked. And he kept this, look at it, you think it's just bits of plastic held together with gaffer tape and some plumbing coming out of it. But it was incredible. And I thought, well, this is great, John, but what do you think we can do with it? He said, well, I know you work on this mouse model of inflammatory bowel disease, and we know that it develops the disease spontaneously and it dies very young. What if we remade that, re-derived it so it was born sterile? What would that impact upon the disease? And I thought, okay.
And he had the inclination that...
He had the inclination that
bugs are key.
The bugs are key to this cause of inflammatory bowel disease.
Yes.
So I was a bit skeptical. My PhD student was a bit skeptical, but we went through the process of re-deriving the mice. It was about 6 months.
Can I just ask, before you say what happened next, what was your perception and your PhD student's perception at the time about the relative importance of the bugs?
Well, I think we had an idea there was some interaction that made these animals more susceptible. We always assume it's an immune defect. So, they've got inherited genetic defect,
right,
and that was messing up their immune system in general. So irrespective of the stimulus, they were always going to behave in this way, and it didn't matter if it was a microbe or something else. So we were more skeptical than John was. So we did the experiment, and lo and behold, the mice survived.
Right.
They were disease-free.
No disease?
No disease at all. They were running around just like healthy.
Really?
Well, they were completely disease symptom-free. And then we thought, okay, we've got to do the other experiment. We've got to take them out of this clean bubble and put them in with it. And then they developed the disease. So that, in my mind, was it. Causality there. Take it away, disease-free, put them back, full-blown disease.
And we will certainly talk about the correlation causation. It was a big theme in your talk today.
Big thing.
At the one Microbiome Health Conference, KTN, so I'm just going to plug it.
I was converted after
You were converted then.
I then became a gut microbiologist, not an immunologist. Well, a little bit, but no, gut microbiology.
And how rare is it for causality to be proven in this field?
It's very difficult. I mean, it's the big struggle with all human diseases associated with your gut microbes. Is it a cause or a consequence? And unless you've got a really good animal modeling, which mimics a lot of the human physiology, the human disease symptomology, it's very difficult to get at that question.
Yes.
So it's hard. And there's maybe inflammatory bowel disease is the disease where we know that the gut microbes are a trigger. We don't know which ones and how, but that clearly we can demonstrate that.
And this highly pioneering work in the sort of cobbled together
the homemade
B&Q lab, which I think is just, you've got to take your hat off to.
Oh, he was way ahead. He was years ahead.
I mean, to have the vision.
Yeah.
So he had an inclination and he went, okay, it's the bugs. So how do we create somewhere with no bugs? I'll just go and build it myself.
Yeah.
That in itself is amazing. And then the fact that he actually managed to do it.
That is incredible.
It's really incredible. Yeah. So when the data started, so I suppose, take us to the moment where you were looking in and going, they don't have disease.
Well, were
you looking at your PhD student going...
Yeah, when the PhD students took me to the animal facility and showed me the mice, said, no, you've mixed them up. They're the mice that they're wild type. They haven't got the gene defect. He said, no, they are. So I made a retest that they had the defective gene still.
And they did.
And that's when I thought, okay, this is real.
Wow. And had anyone done something like that before, to your knowledge?
There were people that were starting to move into this area. In my mind, the godfather of sort of microbiome research, a guy called, his name just gone from me now. Dwayne Savage,
okay,
in a small university in the Midwest. And he pioneered the characterization of gut microbes. He was the one who was first trying to describe what's there.
Really?
And
wow.
This was using classical microbiology tools. So if you can't culture it, it doesn't exist. But he was the first one to come up with a way in which you could start to look at gut microbes.
Wow, I've never heard that name before.
Dwayne Savage. And
I take it he's
no longer with us. He's no longer with us.
Okay.
And then there was a group at the NIH in Washington, Bethesda, that were trying to do germ-free experiments. But they'd taken, let's throw money at it and get a company to build isolators and things.
Yep.
So it was really pioneering days when John had already, 2 or 3 years earlier, started to try and build this thing and test it and make sure it was germ-free.
Amazing.
So
I just love that so much.
And now if you want germ-free mice, you can go to Taconic or one of the other big animal breeders and they'll charge you a lot of money.
Yeah. Yeah.
That was key.
That's a seminal moment in your
seminal moment, yeah.
So that flipped your thinking a little bit. And what were you thinking? Were you thinking, okay, do I do more in IBD or where else does this go?
So IBD was my way in. So with these animal experiments, we could now say, look, we've got clear evidence of a causality of the gut microbes in the animal model. We need to start doing something in humans. So then I started trying to get samples and work with patients, looking at some of the interventions, what was were there any interventions that impacted on the microbiome that alleviated symptoms in the patients? Mm-hmm. And the one that I came across first was this bypass surgery to sort of divert the fecal stream away from a piece of the infected gut.
Yep.
And that caused dramatic relief of symptoms in patients.
So and for the listeners, some surgical procedures involve literally making a kind of loop and having something called a stoma, right?
Yeah, stoma.
So a part of the bowel comes out into the sort of outer aspect of your abdomen. What you're saying, I think, is the downstream part, so basically anywhere below where the stoma comes out, is not getting any bowel contents.
That's right. Yep.
I think what you're saying is they don't have symptoms anymore.
No, because you've removed
the trigger,
which is obviously a lot of the gut microbes there. So you've sort of saved it from that exposure, and it has a chance to heal. And that's, And
was that a by accident finding as well?
Well, I was just talking to surgeons and then sort of looking at how they were assessing the patients and criteria. And then they were telling me about in some patients get alleviation symptoms with antibiotics, which I think, okay, that's another link. So there were sort of these clues along the way. And I thought, well, this is pretty good. And that sort of sent me a little bit more down into trying to do human studies. And again, sort of bringing in immunology as well into that, looking at what impact does all this have on the immune system. Does it suddenly now get re-educated or rebooted to learn not to react to the gut microbes?
What happens to these patients after the bowel gets reconnected? Do they get symptoms again?
They start to get symptoms again.
Right?
Yeah.
Just thinking, imagine if you could retrain just by fasting almost, because if you fast, then it's kind of similar, isn't it? Like you're not.
Well, I mean, if you go with the idea that a mixing your microbiome, you got some pathobionts, microbes normally not going to cause disease, but under some conditions become disease causing.
Yeah, that's what we define as a pathobiont.
Pathobiont. So if you can get rid of those and replace or replenish with healthy microbes, then that might be a more permanent treatment. Mm-hmm. And of course, there are clinical trials going on now, as you're aware, of using FMT, microbiota transplantation, to try and alleviate or treat patients with particularly ulcerative colitis.
Yes. Yeah, I'm aware of 8 studies in ulcerative colitis now.
Yep.
And what's fascinating for me is They're conducted in different locations. There's different routes of administration. So some are nasal, well, basically, I'll call it upper. Sometimes it's duodenal, sometimes it's jejunal, but we'll call it upper or lower. And we've seen a capsule study now recently as well. And despite all the differences in all the studies, there's a signal in all of them. And I'll define signal as some patients are getting much better.
Okay.
And for me, the clinicians in this space will say it's not ready for prime time yet. It's not ready because we need more standardization. We need to know how many to give. And I get that. I really do, because it's not a licensed product and you're taking a risk. But at the same time, I'm thinking, well, hold on a second here. The fact that there are some patients doing so much better, and a few do get a flare up, but that seems to be the minority rather than the majority. Should we not be giving more patients with ulcerative colitis FMT?
So I think you need to turn that around and say, why are those patients not responding?
Yes.
Because, we anticipate a successful outcome.
Yeah.
So why isn't it all patients getting a benefit? What is different about those that are not responding? And my understanding of ulcerative colitis is it's different clinical subtypes, different symptomology, be it severity or qualitative difference in the symptoms. So I think we need to marry much better the intervention to the clinical phenotype, because then I think you've got a better chance of having success across all patients with that phenotype.
Yeah, no, I'm with you, and I think the phenotypic characterization needs to be a microbiome profile as well.
Yes.
For really being futuristic. One of the other things about UC is it an inducer of remission, or is it some maintenance element, or is it both? Do you give a really intensive regime like the Australian study by Parham Sothe where they gave like 40 FMTs?
Jeez.
Yeah.
It's a lot.
One colonoscopy and then once a week by enema every week for 8 weeks, I think, or something like that. I need to get my maths right. I've got my maths wrong. I won't embarrass myself any further on this podcast, but in my mind, I think it's something like 40 FMTs. And then there was another one recently that combined healthy diet with FMT as a maintenance, so maintain remission. And I think both have a place, right?
Oh, absolutely, because if you want to sustain your engrafted healthy microbiome, the best way to do that is to feed it the right things. Yeah. And that's your diet.
Yeah. So when someone says to you, what do I need to do to make my microbiome better? The first thing that springs to your mind is probably diet, right?
Yeah. And that actually is the first line of treatment for inflammatory bowel disease, isn't it? You start removing things that you think might be a trigger for overactivation of those pathobionts. Yeah. So the first line defense is, okay, let's go to, eventually you end up with an elemental diet, parenteral-enteral nutrition. Yep. And that can be beneficial. If that doesn't work, then you go to antibiotics, steroids, biologics, and then the last resort is surgery. Yeah. So we're starting off actually at the right end. Yeah. Let's try and change it through diet and or knock out the microbes that we think are responsible. Yeah. Yeah. I mean, in kids, it's phenomenally successful for first diagnosis. Yep. And I was speaking to someone called Richard Hansen, who was the first guest on this podcast last week, just catching up because he's taken up a new role. And he was saying that actually the paradigm's changing a little bit in IBD, and they're going in with a immunosuppressant and dietary intervention at the same time. Sledgehammer, yeah, to try and really just knock it on the head. So I thought that was really interesting. But he's firmly of the view that you need to combine FMT with dietary manipulation. Yeah, I would agree. I kind of agree with that, right? I would, yeah, I do. Yeah. If you want to make a lifelong commitment to change your microbiome, I'm sorry, or I'm not sorry actually, because you'll feel good, but you have to make a lifelong commitment to change your diet, right? Yeah. Or could you have an FMT from someone who's made a lifelong change to their diet? Yeah, the gut-brain axis. Okay, I wondered when you were going to bring it around to that. Yeah. Well, there's something here, because there are good examples, some examples of food preferences being influenced by alterations in your microbiome. And the one I'm most familiar with is actually chocolate or meat. The smell, the taste, the texture. After an intervention, it changes perception. sensory perception of the food you eat. And that can be a good thing or a bad thing. And in a lot of cases, it's, I no longer want to eat that because it smells bad, it tastes bad, and it's just, the texture is just yuck. So I'm not going to eat it anymore. And that was part of their staple diet prior to the intervention.
The FMT?
FMT, probiotics, prebiotics, things that would shift your microbiome. The best example I know is gastric bypass surgery, where these preferences have been documented in patients pre and post bariatric surgery for weight loss. Suddenly their food preferences have changed without any conscious effort. It's just, no, I don't like the smell, the taste, texture, and they don't eat them. And meat and some dairy products are on that list.
Can we talk about the chocolate example just briefly?
Chocoholics, yeah.
The chocolate.
So again, so they go to eating.
Well,
it's interesting because there's also people that, and these are sort of case studies, so it's not by any means scientific rigorous. Yeah. But there are people that really like dark, strong chocolate, 70, 80% cocoa content.
Okay.
And they used to consume lots of it. Post-bariatric surgery, that was just, too bitter, or now it was just completely unpalatable.
What?
And they stopped eating it. And then there are cases going the other way. So, eating lots of white chocolate and stopping altogether, dairy milk chocolate, whatever, they would just stop eating it because it no longer gave them that pleasurable sensation. So had the treatment interfered with, the gut hormones, serotonin, the pleasure hormone, that would be the smoking gun, right? That you've changed the balance in all these hormones that influence behavior. Serotonin being the key one, because most of the serotonin the body is produced in the gut.
Yeah. Wow. Okay. So basically, if you love chocolate, could be because
your gut microbes are telling you we like it.
They like it a lot. We want more. And they're producing, is this us now on subject of BEVs?
Not yet. No, not yet.
But they're producing, the listeners probably like, what are they talking about?
Well, they're influencing the balance of hormones. So most of your hormones derive from specialized cells in your gut lining it's all part of the enteroendocrine system. So there are specialized epithelial cells that line your gut that make hormones and neurotransmitters as well. And we now know that there are populations of gut microbes can actually make them as well as the body making them.
Really?
So there are a lot of bacteria that can produce a neurotransmitter from an amino acid in your diet in a simple one enzymatic step reaction. So it's not complicated biochemistry here. It's one enzyme acting on an amino acid precursor to make serotonin, GABA, histamine. So you can imagine if you've got a lot of bacteria in your gut that can make histamine, you're likely to be more susceptible to hyperallergic reactions to certain foods. So
is that what causes anaphylaxis? Maybe
it can certainly contribute to it because you can sensitize yourself to an allergen on your skin, and then if you eat it, the reaction is even more violent and more rapid because again, the mucosal cells have gone to the gut and they're primed and ready, and you consume it and bang, anaphylaxis if you're not careful.
So can we sensitize or improve people's anaphylaxis through the microbiome somehow? This is new for me. I'm not, this is a new conversation.
It sort of goes beyond describing what's there to what's being produced. So I think you'd have to be measuring the content of these things in a stool sample. So what we call the fecal water. Yeah. You'd have to isolate the liquid phase of your stool and analyze that for these hormones and neurotransmitters. Do you have more of one or the other?
And getting a sample of the liquid stool must be hard, right?
It's not that difficult. I mean, you just take a stool sample, you put it in a centrifuge, pellet all the hard matter, the solid matter, and you just take the upper liquid phase, filter to sterilize it, and then off you go.
So sorry, I thought it was more like you have to get what becomes the stool in your GI tract rather than isolating the stool.
So that's good, because I'm glad you asked me that, because the health warning with all microbiome studies and what we're describing is it's based on a stool sample. And we're assuming that accurately reflects the population's activity that occurs in your gut. And that's not true. So we have to be careful, but it's a fact that's often ignored.
But it must still be very useful though, right? Because it's like the end part of all your digestion.
Yes, but if you consider where microbes live in your gut, we now know there are different populations in different regions of your gut. And those that are attached to the mucus layer that lines your gut are less likely to be in your stool sample. You're going to be looking at those that live in the lumen of the gut, in suspension everywhere.
So for the listener then, how do we help them understand all the various microbiomes in their gut? So there's the luminal microbiome, which is like the space in the middle. And then there's your...
So they're the population of microbes that live on top of the mucus barrier.
Yep.
It's a thick, sterile layer of mucus. It's like a gel-like structure that covers the boundary cell, the cells that are the entry point to your body, your gut epithelial cells. And so that's sterile, so it protects from direct contact with gut microbes. But there are microbes that love to feed off the material that makes up the mucus, so they will live on that outer layer.
Yep.
And of course, unless you have diarrhea or food poisoning, you're a lot likely to displace them so much that they'll start to appear in your stool sample. So what you're looking at in your stool sample is the microbes that live in the liquid phase of the lumen. They're attached to food particles that clump around food particles. They will appear in your stool more frequently than those on the mucus.
So how do we analyze the bacteria in the mucus?
Biopsies.
Biopsy.
Colonoscopes where you can go in.
So it's tricky. You've got to physically scrape off. Yeah.
You've got to basically pinch off.
That makes sense. Yeah. And then that's a very specialist.
It is. Yeah.
So the stool is the best we can do without being very invasive. And
I mean, it's very good, but you just have to bear that caveat in mind.
That makes sense.
It's a surrogate.
That makes a lot of sense. And of course, within the stool, there's many different things, right? So we've been talking a lot about bacteria.
We have indeed.
But we should maybe just explore the other domains.
Yeah, so this is one of my pet peeves, is when people talk...
I'm glad we're on that, yeah.
When people talk about the microbiome, it's always about the bacteria.
Yes.
Oh, it's like, everything begins and ends with bacteria. Yes. But it's not.
No, it's not. No.
We have gut microbes, they're predominantly viruses. There are equal numbers, if not more, viruses than there are bacterial cells. And then we have fungi, which make up part of your normal microbiome as well. Ancient bacteria, which are a cross between human and microbe, which is called archaea.
Yep.
And then there are protozoa as well. So it's a complex community of different things. But we've just focused on the bacteria because they're easy to analyze, they're easy to identify. They've got unique signatures so we can identify them and then classify them. Viruses don't have that, so they're harder to describe.
Yeah, because is there any evidence that actually proves that the bacterial element is the most important?
Well, I mean, I could say everything that's been published on gut microbiome and health is related to the prokaryote principle. So the beneficial effects that have been described are associated with the gut bacteria.
Right.
But there are large numbers of viruses that live inside bacteria and can change the behavior of bacteria. So a good question is, are the changes we're seeing in gut bacteria due to the effect of viruses, which are invading, infecting, destroying, giving them new functionality?
Yeah.
So are we just looking at the effect of viruses when we're looking at bacteria?
Quite possibly. I mean,
so it's possible.
I think part of the context for my question there was, well, as you know, I'm working in the industry side, and I'm leading a company that is of the view that the ecosystem as a whole is more important to look at than any one constituent in most cases. Now, clearly there's examples that you can give. So candida infection in someone who's immunocompromised, which is a fungus for the listener. C. difficile, you have overgrowth of one pathogen. But another more complicated diseases, maybe it's an interplay of the prokaryotes and maybe even the eukaryotes as well. And where I was going with this is that there's a lot of companies who've decided that it's the bacterial component that's the most important. And we hear a lot about defined consortia, and we're going to move to something more defined and characterized, but do we actually know enough at the moment to be able to say, yes, it's going to be a defined cocktail of 3 or 5 bacteria? Because the microbiome in all of our patients is so different, right? So why do we not want more shots on goal? And why does it have to be defined?
Yeah.
See what I'm saying?
Yeah. I think it's a big oversight because it doesn't begin and end all with bacteria. That's clear. And, I think there is a slow realization that we need to look at the integrated microbiome to understand it because, they're there for a reason, right? I mean, it's not just bacteria there. There are other components. So clearly we've decided they're important to retain. We don't want to get rid of them.
Yeah.
So they must be doing something. And, they've reached an equilibrium with all the bacteria. And so there's mutualism there, there's symbiosis, there's mutual benefit to all of them being present.
Yes.
Whether or not some are providing food for others, some are providing a protective niche for others. It's a very complex ecosystem. And somebody said to me the other day, we know more about the surface of the moon than we do what's living in our gut. We're just scraping the surface and looking at the bacteria. There's a huge amount.
When you put it like that.
Yeah. And we don't know much about the surface of the moon.
No, we don't. We need to get to work.
Yeah. So it's
so much still to learn. That's crazy.
So, in terms of, is microbiome research done and dusted? Absolutely not. We're at the beginning of this, and it's a long journey before we can say, okay, we can understand. I covered this in my talk today. We understand only a fraction of the factors that influence why your microbiome is different from mine. Lifestyle and behavior, yes. Okay, medication, yes, we'll tick them off. But that's less than 20%. So what is the other 80%? And that has to be, the nature of the interaction between all the components. Things we've got no idea, well, little insight as to what they are. But they are the big determinants of what your microbiome is and what it's doing and how it's keeping you healthy.
Sorry, can I just... The 80% of the factors are currently unknown.
Yeah, so this comes from 2 very large population-based studies in Belgium and the Netherlands, thousands of individuals.
Yeah.
And they've looked at, okay, what factors contribute to differences in the microbiome? And, they ranked these things. And at the top, not surprised, medication. So,
and that includes antibiotics.
Antibiotics,
yeah.
And then
there's others, right?
There are others, yeah. Your blood, the composition of your blood.
Your blood type.
Factors in, well, not blood type, I think it's composition and like blood chemistry.
Oh, okay.
Then obviously there's a lifestyle, where you live, how you live.
Farm.
Diet, yep. Farm, city. Do you live in shared dwellings? Do you have pets? All these things, fact, do you have children? Cultural differences. You take all of those together and it counts for less than 20% of the variance. So that is the rest, is coming from within. It's the microbes deciding who's beneficial, who's not, who they need to keep, who they need to get rid of. So it's that black box.
And does that bring us back to what you were talking about early on in this conversation around the early years being so important?
So, yeah.
Because that core group probably says, actually, no, you're not allowed in here.
Yes, so that's really important. So if we look at what we know about how the microbiome changes with age, clearly the first few years of life are critical because it's then building up, it's reaching its sort of life, its point at which it's not going to fluctuate much.
Should we be doing something during those early years?
Absolutely. I would argue if you want to change the microbiome, it should be done in the first 3 or 4 years of life.
Wow.
That is the best intervention.
Wow.
And the things that will promote the development and establishment of a healthy microbiome are how you're born, where you're born, and how are you nurtured. So, vaginal delivery, cesarean delivery, hospital, home, breastfed, bottle fed. Those are key factors in the establishment of a healthy microbiome. And, we can enhance that by providing to those individuals who don't get the benefit of those beneficial things occurring very early in life with the microbes they're missing. And that should be lifelong. If it's done in the right way, right number of intervals, right dosing, et cetera, right mixtures, you should be protecting people, that individual, lifelong, because they should be stable and carry with them through their life.
The challenge and there's many challenges, but I think the key one must be, even if you develop this beautiful supplement or drug, proving...
It's a long-term study, isn't it?
Yeah, so I knock on the doors of investors all the time. I say, hi, I'm James, and I've got this great idea. I think it's going to change the world. We've got some good data, and here's the trials. And if I was to go into an investor and say, well, I've got this is going to be big, right? Everyone's going to get this if they were born through a C-section or whatever. And there's going to be less allergy, less obesity, less autoimmunity, but I need you to buy into me doing a 50-year study.
Yeah.
They'd all be going, no, we can't do that. Yeah. Most investors want a return in 5 to 10 years. 10 years is really pushing it. 5 years is more standard.
And there might not be a tangible
endpoint. Yeah.
So there is a benefit to being in a research institute where you have that, Quadram, where you've got sustained long-term investment and the support of, a stable funder, like the BBSC, for example, in our case, where you can say, this is our strategy. To do this, we need to undertake these studies. And, we need as long as it passes peer review and everything else, they will buy into that to allow us to do that.
You need the patient capital.
We need the patient capital, exactly. So that's one way you can do it. But again, not everybody works in an institute has the benefit of that sort of long-term sustainable. And of course, that allows you to retain key skills, people that can do this, highly trained, so you've got confidence you'll be able to get some end product.
So what is your research focused on at the moment? And if I have missed any key part of your journey, let's go back there and let's just explore that before we talk about the now. And we've got lots of difficult questions to touch on as well still.
So when I went back to the UK around 2000, I went to Leeds because there was some bacteriologists there who were really interested, a guy called Keith Holland, who worked on acne and the bacteria that causes acne. And he was very helpful to me because he worked on a very difficult to culture organism. And I wanted to work on an anaerobic bacteria that grow in the gut. So he worked with me for several years, 3 or 4 years, to basically build up the toolkit so that we could start working with difficult to culture microbes in the gut. And then unfortunately he retired, and the university in their wisdom decided they were going to hire structural virologists rather than classical bacteriologists.
Okay.
So that was the time that the Institute of Food Research, as it was then, came knocking on my door, said, would you be interested in developing a gut health program? And I knew they had a lot of really good microbiology, basically in the food side, working on lactobacillus, probiotic bacteria, but they're also working with anaerobic bacteria. What they didn't have was more the gut side, the humans, the host side, if you like.
Right.
So when I went down to IFR, we sort of built a program around gut health, and it was microbiology, host health, immunology, etc. And that's then evolved into the Quadram Institute's programs since then, where we've now got sort of good strength on both the host side and the microbe side. And the microbe side is looking at fungi, bacteria, viruses as well. So we've taken the integrated microbiome approach. So that really changed the Institute of Food Research into more fundamental gut microbiology, gut microbes and health program, which it is now. But I never lost my own personal interest, which is, how does the microbiome work and how does it interact with us, the host? And it's not through direct physical contact, so we're not constantly acquiring microbes from the gut and then processing whatever. Yeah. There's that sterile barrier. So what else can get across that? And
so can I just press you on that a little bit? So that's the sterile barrier. So, and I'll ask you in the context of FMT, because I'd always thought that in an FMT, and for the listener, fecal microbiota transplantation, if you've Not heard of it, and this is the first time hearing it. Go to Biome Bites episode number 1, where I talk about intestinal microbiota transfer FMT. So I thought that when you instill this, I'll call it ecology, the immune system recognizes it as being foreign. But I think what you're saying is only if it comes into contact with cells that are innervated or immune cells, right? And they're not going to because of the mucin.
Correct.
Huh.
Yeah.
But some of them might find their way. So
that's always the danger because, like I said, there are people out there that believe all human diseases are related to changes in your gut microbiome. And so how does that work? You've still got to communicate, with the host and drive these nasty responses. So I think the underlying cause is what we call a leaky gut. There are breaches in the barrier.
And that barrier is the mucin.
The mucin.
And the epithelium.
Yeah. Or even worse, if you're disrupting both your epithelium and the mucus at the same time. And this could be through, drugs, medications.
Alcohol.
Yep. Yeah, these things over a long period of time will make the barrier leaky.
Yeah.
And that then, depending on what's getting across the leaky barrier, could then trigger all sorts of nasty reactions. And the gut is a massive sensory organ. It's not just got the biggest immune system in the body. It can also make virtually all known neurotransmitters. It's your second brain. It's packed full of neurons, the enteric nervous system, and it's packed full of hormones. So it can produce all these hormones.
When people say gut feeling.
I get this gut feeling. Absolutely right.
Gut instinct.
You get the butterflies in your stomach. That's all these neurons in your gut firing off. And that could be to say, fight or flight, or stop doing what you're doing because this could harm us.
Wow.
So depending on you stimulate your immune system, your second brain, your hormonal system in the gut, you can get different signals going all over the body.
Wow.
And of course, a lot of them will end up affecting the brain and maybe how you behave and respond. So it's a massive sensory organ.
This gut-brain axis then, should we just touch on that briefly?
Yeah.
So it's a bit of a buzzword at the moment, isn't it?
It is.
What does it mean to you?
So classically, it's the connectivity between the brain and the gut via a major nerve called the vagus nerve. And that's a bidirectional nerve. So the gut is communicating with the brain is communicating with the gut, usually to regulate things like peristalsis, motility, and the brain is signaling via that nerve to say, we have a problem, we need to change peristalsis, we need, diarrhea, for example, you get a massive brain causing massive peristalsis and trying to get rid of the toxins. And it's feeding information to the brain about what's going on in the gut, basically. So the vagus nerve is there anatomically.
So that's a key element.
That's the key element. Direct connection.
Yeah.
And then you've got the other systems, so through the immune system and the vasculature. So if things get across the gut into the bloodstream, they can then circulate around the body, get to the brain, cross the blood-brain barrier, influence the brain activity. Hormones, of course, neurotransmitters, they'll all work through the nervous system, other nervous roots to influence the brain. And now, of course, it's, what's triggering all these reactions to stimulate these pathways?
Right.
And we now think that a major part of that is your gut microbes, what you have there and how they're interacting with the nerve cells, the hormonal cells, the immune cells to trigger these things. And as I said earlier, we now know that gut microbes can make their own neurotransmitters, so they can directly influence the brain if those get into the bloodstream.
What triggers them to make them? Do we know?
We don't really know. We know that sometimes it's in response to stress.
Wow.
So it's interesting because some stress hormones, for example, acetylcholine, can be utilized as growth substrates for certain types of bacteria in your gut. And interestingly, salmonella is one of them. Huh. So, there are microbes which can act in response to the hormones we produce. And then in response to various stimuli like an infection or something, that will cause bacteria to make neurotransmitters or hormones to change this. So there are probably a variety of stressors that can trigger this production.
Incredible. So really, it is a fair statement to say, trust your gut feeling, gut instinct. I'm gutted. Yep. So it holds down back to the gut.
Not far off the mark there, actually. Yeah.
Amazing. Good. So back to your work then. We mentioned a little bit about leaky gut. We mentioned communication between the microbes and the body. So how are you answering the big questions that you described? Like, what is it and why is it important? And that kind of thing.
So what are the mediators of normal communication between your gut microbes and the body? And like I said, there is this barrier. So we know that it's not intact microbes which can trigger the reaction. So it has to be small, diffusible molecules like nutrients, hormones, but also extracellular vesicles are part of this. So extracellular vesicles are made by all cells in our body, and all our microbes produce them as well. And the ones that we work on bud off from the surface of a particular type of bacteria or category of bacteria called gram-negative bacteria. They bud off the cell wall, and these form microvesicles. And these are very small. They're about 100 nanometers. So they're approaching the size of a lot of virus particles, for example. So they're very small. And they can permeate through the mucus. And we know that they can then get taken up by the epithelial cells, the barrier epithelial cells. And depending on what's inside the vesicles, they can change the behavior of those cells. And we now know that they can cross that epithelial barrier to access cells beneath them, which are mainly immune cells. A lot of immune cells live below the epithelial layer. And so they can influence the immune system. And that can be a good or bad thing depending on, again, what is the cargo they're carrying and what type of bacteria they derive from. So there are a lot of pathogenic gram-negative bacteria, like Salmonella, for example, is a good one. Enteropathic E. coli, which utilize their vesicles like a Trojan horse. They will put toxins inside their vesicles, and they will then be taken up and they will damage.
Ship them out.
The epithelial cells ship them out, causing a breach, which then the parental bacteria can enter through. So that's a pretty clever strategy. So the question is, well, commensal bacteria, the good bacteria, don't want to do that. It's detrimental. So they get acquired through natural sampling pathways that epithelial cells use to sample what's in the lumen, decide whether or not it's harmful or not.
Yes.
So they can utilize that to get inside epithelial cells, cross the epithelial cells, and to interact with immune cells. And so we call these bacterial extracellular vesicles, or BEVs for short. And all bacteria produce them. A lot of cells in our body will produce them. They're called exosomes.
Yes.
And they circulate around the body. There's a lot of interest in those because they potentially have therapeutic value, particularly as anti-cancer treatments. So there's a lot of interest on these extracellular vesicles produced by the cells in our body. But we focus on
the work
the bacteria.
Before we get into the technicalities of BEVs, can we try and talk about what a healthy microbiome? You knew it was coming.
I knew it was coming. Yeah, I knew it was coming. Well, the simple answer is, if you're healthy, then you've got a healthy microbiome. But it's a difficult question to ask because all our microbiomes are unique.
Yes.
Now, we do know there's a core microbiome.
How unique?
Well, there's arguments that we can use our microbiome as our microbial fingerprint.
Right.
And our microbiome can distinguish maybe 90% of us. And it can distinguish identical twins. So genetically identical twins will have a different microbiome.
Wow.
So, they're clearly, it's not all us deciding on what microbes will populate our bodies. The microbes are influencing themselves and what else other ones will populate our bodies. So, as I said, there is a core, and it's about, we believe, 50 types of bacteria that are present in 90% of all of us. Not surprisingly, these bacteria provide essential complementary functions. A lot of them are those that produce essential micronutrients, so vitamins. Yep. Vitamin B12 is a good example. Our bodies can't produce vitamin B12. We rely on the production by our gut microbes or what we require from our diet.
Yeah.
So there's this core which allows us basically to digest and process plant-based materials in our diets.
Is it fair to say that those core groups must have been on the planet for so long before us? Because we've all evolved to kind of need these core groups, they must have been on the planet as we were evolving, right? Or
yeah, it's just this is the chicken egg argument. So which is the most important? Did the microbiome, help us evolve as to we are? Certainly in terms of our capability and ability to adapt to different food sources, which is the ultimate thing we need, right, to sustain life. And then we just provide a niche for these microbes. So did they exist in these complex communities before we came along? That's an interesting question. So these communities probably did exist before. So question, where did we acquire them from?
Yeah,
and my belief is that a lot of them are aquatically derived. So as
interesting.
Animal evolution, we moved from the water to the land.
Yeah.
We and others have identified genes in our gut microbes which we can track all the way back to aquatic microbes.
Oh, wow. From the vents?
No, this is just freshwater, seawater.
Really?
Some live on seaweed, for example.
Oh, wow.
The Japanese population, in their microbiome, they have a high proportion of a particular enzyme and microbes that produce an enzyme to allow them to digest seaweed in their diet. So it's this self-fulfilling. So, you want to eat lots of seaweed, you need the microbes to process it to get the health benifits.
I love sushi.
If you don't have the microbes, well, you better make sure you got the right microbes, get the maximum benefit. And it's the same in our work. So the microbe we work with has enzymes which we can track all the way back to bacteria that live in oceans. So it's an idea, but it's interesting that, these communities may have evolved as we, from the water to the land transition, and they were around at the time we evolved. This is, because how you prove it, I don't know.
No, we need to talk more about this. I love these out-of-the-box theories.
It would sort of map nicely onto
interesting.
What we know about human evolution.
But I just find it so fascinating that this 50 seems to span the whole planet.
Yeah. And that is what I think is your healthy gut microbes. That would define healthy gut microbes.
What order of classification are we talking about now, to get a wee bit technical?
Well,
genus?
No, we're getting down to strain level.
Whoa, really?
With the technology we now have, we can get down to, brothers and sisters of the same parent. We're down to that level.
Yeah.
So there are groups at the Quadram that are very interested in defining strain level, and where do those strains come from? Do they come from our parents, our grandparents? Where did we acquire them? How far back can we go?
It would be fundamentally transformational if we could prove that it was this group of 50. Every human on the planet had some, let's just call it 70%, or they had 30 out of the 50.
Yeah.
Doesn't matter what they are, but if you've got this 30, you're going to have the functionality you need. That would be, and the rest is just, I guess they're almost like passengers. They're no, they're not.
It's more that, so what the core is, it allows you to survive, to be able to survive on the diet that we've adapted, we've evolved to consume. So that's core functionality, core survival.
But not optimal.
How well you survive, how long you survive.
Yes.
That's, all the rest. Yeah. And that's when, lifestyle, behavior, habit, where you live, your genome comes into play. So, the core is there to keep us alive, sustain us. Yeah. But then how well we live, how long we live, that I think is where everything else comes in.
Well, let's jump into that. But before we do that, I've got a bit of a confession to make. I think I'm addicted to sushi.
That's not necessarily a bad thing.
I eat so much sushi, and my theory is that there's something in my gut telling me to eat sushi. So I don't know. Is it some helminth or something like
No, it comes down to cravings. And
I get cravings for sushi.
Yeah, I mean, some people get cravings for chocolate. I mean, yours is probably a more healthy. More healthy one.
Don't eat chocolate.
So it's like, there are signals being delivered to your brain saying, you need to eat this more because it's good for you and it keeps us there. So it'd be interesting, if you had an FMT, would you still have a sushi craving? If that FMT donor never had sushi or didn't like sushi, would that change your craving?
No, but I love sushi, so I can't have an FMT and change it.
Okay, well, you better pick your donor carefully then. Yeah.
Well, as you know, we're mixing together microbiomes at Enterobiotics, so maybe they cancel each other out. I don't know. What you're saying about cravings is really interesting. I'll just I'm quite familiar with the work in liver disease and FMT, and there's this researcher called Jaz Bajaj based in Virginia. And what he's shown is that alcohol cravings reduce after FMT. And he's shown it pretty convincingly in heavy alcohol.
Any particular type of alcohol? I mean, beer, spirits, wine?
I don't know the answer to that. I think that's a really good... I mean, alcoholics typically would just drink anything.
Yeah. Usually high alcohol spirit.
Based on my experience, not as an alcoholic, but working on the alcohol rehabilitation service. And they would drink. They'd have a staple, but it's just about high consumption. But anyways, in this study, they did the FMT and the cravings reduced significantly. He is a pioneer in the gut, liver, brain axis and microbiome modulation and manipulation.
Yeah.
And what I'm hearing is that these cravings may be microbial driven by the microbes.
It's impossible. Yeah, it's possible. Yeah.
Right.
Because in alcoholics, with every person that eats a specific type of food, your microbiome is going to adapt to that and realize to survive, we have to use these as substrates.
Yeah.
We have to feed on them. So you'll get shifts in your microbiome to promote the growth of those able to utilize whatever it is you're now consuming in vast amounts.
So maybe it's adapting for the alcohol.
It's adapting. And we talked about Harry Flint earlier, and he did some pioneering studies looking at how quickly the microbiome adapted to interventions, dietary changes, and it's within 1 to 2 days, you see a shift.
Wow.
And then, of course, if you revert or go off the diet, again, you get the shift and you can get the rebound effect. And from a weight reduction period, that's where some people experience this massive weight gain after coming off a diet, because you've got this rebound effect in your microbiome. Yeah. And suddenly it's exposed to everything it used to have. It's energy dense and it's busy, depositing lots of excess energy into fat, et cetera, et cetera. Yeah. So, your microbiome learns. It's a smart system.
Incredible.
It learns to adapt to whatever it has to, because it's a survival instinct, if you like.
So I guess when someone says, Professor, what do I need to do to change my microbiome and how do I do it quickly?
Well, my advice, actually, it's the government's advice. You need to get a minimum of 5 servings a day of fruit and vegetables, the colourful plate. And that actually, it might sound trite and simple, but it is actually good advice because it contains everything you need to keep your gut microbes healthy. So you might say, vegetarians, they're going to be the healthiest people on the planet. Well, in some ways they are, but they do suffer or can suffer from insufficiencies, vitamin insufficiencies, because the meat in your diet provides a good source of key micronutrients, which when meat's not in your diet, you need to take supplements.
Yep.
So a lot of vegetarians suffer from vitamin insufficiency.
Fascinating. Absolutely fascinating. I love the fact that the government health advice,
they got it right once.
They've got it right. The government health advice is microbiome health advice.
Interesting, though, not evidence-based. So they knew not at the time that advice came, they knew nothing about microbiome.
There we go. This brings us back to the.
This has to be healthy.
This is it. People, to be fair, if we're at the conference today, we could take. It's a microbiome conference. A really bad example. We'll go to Buchanan Street in Glasgow, and we're going to take 100 people, and we're going to ask them all, right, what do you need to do to become healthier? Now, we are in Glasgow, and we have very, very bad public health in Glasgow.
Deep fried Mars bars.
I'll be optimistic and glass half full. And we said, right, what do we need to do? I bet you they would all say something along the lines of, well, stop drinking so much. If I'm smoking, stop smoking. I need to eat more fruit and vegetables and do some exercise and go outside a little bit more and sleep more. Okay. Right. So how much of that is having an impact on your microbiome? Probably all of it. But the thing that's most impactful is what we're eating.
Yeah.
So this health advice is microbiome advice. So maybe the microbiome is health. Maybe the key to our health is the microbiome, as we've been discussing for the last hour and 10 minutes.
It is. I think it is. Yeah, it is key to your health.
And how, we talk about 5, what if we have 10?
Well, this is the sad thing.
What about 20?
There were surveys have been done to show that there's only 20-25% of the UK population actually getting 5 servings of fruit and vegetable a day. And in fact, there's been government missions to try and double it.
Yeah.
So that is clearly
it's not worked.
Pipe dream. because we haven't got enough people just consuming 5 servings. And the other thing, of course, is dietary fiber. So, there are all these recommendations about you need this level of dietary fiber.
Yep.
And I looked at a survey maybe a year, 18 months ago, and there isn't one country in the world that is reaching the recommended intake of dietary fiber. Even you think of the healthy countries, there isn't
Japan?
one country. No. They all fall below their national and the WHO standards. So if we want to improve world health, there are simple things you can do. Just increase dietary fiber intake, get more people to eat fruit and vegetables.
I'm equally stunned and perturbed by that incredible fact.
Well, I didn't believe it either, but it is true. I mean, this came from the WHO sources, this information. And there's lots of countries that set up initiatives to try and get to that level.
Goodness gracious.
So one question, of course, is, well, what's that level based on? What's the evidence that, you consume this number of grams a day? And, it's a bit flaky. So, what governments may do is say, well, actually, we'll lower them a bit. And then we can say,
we're hitting our target. No,
50% of the population now are doing this. When, a lot of people in the know, it's actually, no, we need to be increasing this even higher. So, the science is going one way, and I sense politicians and everything.
So what do we do? Subsidize fruit and vegetables for the masses? Oh, can I just plug enterobiotics, please? Can I just indulge myself a little bit? It's unlimited free fruit and vegetables for everyone at Enterobiotics.
Is it?
And that's a fact. Yeah. And I'm proud to say it. And also the fresh pressed green juice. It's unlimited for everyone.
Yeah.
We've got a Vitamix in the office. And, it makes me very happy to say that. Now, I don't know how many people within the company are hitting their fiber intake.
No, I mean, at the Institute, we used to have coffee and biscuits in every meeting. I said, no, we'll have fruit and coffee.
Nice.
So
nice.
And being the Institute of Food Research at the time, you think, well, this is what we should be doing, right? Yeah.
Walk the talk. Okay. But what could we do? What about a wee sachet that contains loads of fiber? Just put it in your coffee. Or is that too simplistic?
It's too simplistic, and you're missing the benefit of eating the intact fruit and veg, which has other nutrients in it as well, micronutrients in many cases. You distill that down to a powder, you're losing. It's like when you overcook vegetables, you're losing a lot of the beneficial effects of it because you've destroyed them in the cooking process. So eating raw fruit and vegetables is the best way to maximize the health benefits of fruit and vegetables.
Should we be washing our vegetables?
Good question. I think if you know where they come from, in supermarkets, are very cautious about not having contaminated fruit and vegetables on their stores, obviously. Yeah. So there probably isn't. But, in the SARS-CoV-2 pandemic, a number of people that were wearing gloves to handle things and then going home and washing things and, wiping their hands were all continuous with alcohol swabs and things. It's too excessive.
Right.
So, I mean, you can wipe the surface of fruit, but, to wash it, I think that's probably not required. Particularly if you know it's coming from, one of the big chain supermarkets.
Yeah, that makes sense. Right. Let us go back to the healthy microbiome. How important is diversity, in your opinion?
Yeah, so we hear a lot about this, that the altered microbiome in disease state is principally due to a loss or reduction in diversity. In a bigger context, we need a diverse ecosystem. We need diversity in plants, animals. So diversity is good.
Yep.
But I would argue in the gut microbiome, if you've got the core microbiome and, your functionality is, you're healthy, you're fit, does it matter if you've got a diverse microbiome? Probably not. So again, it's how we translate, all this taxonomic data we get from sequencing to actual phenotype and function.
Why is it that across all the case control studies and even prospective longitudinal studies where we look at people as they progress from sick to more sick, we always see the same thing, which is there's a reduction in diversity between disease and control. And as someone gets sicker, they get less diverse. So
sicker people will have more medications, their diet, they're probably not eating as well as they could.
Okay.
Of course, all these things are going to compound.
Yeah.
And don't forget, every drug you take, your gut microbiome, see, it's food. It's something
drug.
they can process. Every oral medication you take, and 80% of our medications are oral, right? Pills, tablets, liquids. Your gut microbiome is another source of food for them. So they will metabolize your drugs. Now, that can be good. So like for metronidazole, which is a prodrug, for that to work, it has to be processed by your gut microbes to make it effective. But there are other drugs, if they get metabolized in the wrong way, produce toxic factors, or they completely inactivate the drug. So, you may have to give more. And the good example of this is cancer therapeutics, right? There are cancer drugs which work very well if your microbiome is a certain type. If you don't have the right microbiome, then it's not going to work very well. And there are antiviral medications that work in the same way for treating vaginal viral infections. If your vaginal microbiome is a certain type, the drug will work very well. If it's not, you don't have those key microbes there, it won't work, or it could even be worse. So, everything you consume is going to get processed by your gut microbes, drugs as well.
Yeah.
And so it becomes important, you can see this becoming important in terms of treating individuals, not groups of patients, to look at their microbiome and say, it will work in yours, but it's not going to work in yours because you've got the wrong type of microbes. So instead, we'll give you this drug. So instead of this, panacea, same drug, everybody, you're actually now personalizing this, personalized medicine, but it's microbiome based.
It's so interesting that basically everything you put in your mouth probably has some sort of impact on your microbiome and vice versa.
Yeah.
I was out for dinner in January at this medical conference. It's actually a biotech conference in San Francisco. And the person I was with was like a very traditional drug developer. And he said something along the lines of, I've got no idea why people take turmeric and all these oral supplements that have potential impacts on the wider body, because none of it gets absorbed. And I was like, well, hold on a second.
Doesn't need to be.
I was like, you're missing something so big. It doesn't need to be systemically absorbed because it might be absorbed by your microbes.
Yeah.
And then they produce things that are then pushed into the systemic circulation.
Yeah.
And what I took away from it, it's a super, super smart, successful person. What I took away from it was, I'm not sure the pharmaceutical industry yet understands that many of their best-selling drugs could sell much better if they understood the microbiome that it was going into.
So I think it goes deeper than that. So can you tell me which medical school has microbiome science as part of the medical curriculum?
Well, I went to medical school, and guess how much microbiome was on the curriculum?
Probably not too much.
None.
Yeah. I don't think it's because I teach medical students at UEA.
But I think it's changing.
Yeah, it's not where I am, because I've continually advocated, we need to educate these medical students about the importance of the microbiome.
We have to. Well, so to be honest with you, going back to day one of enterobiotics, 24-hour computer laboratory, running late to select a topic for my dissertation. I was doing a very nerdy form of procrastination, going from medical journal to medical journal. I arrived at the Nature paper from Jeffrey Gordon that showed if you move a microbiome from a thin mouse into an overweight mouse and overweight mouse into thin mouse, you see weight loss and weight. And I was just like, what? Like, what is that?
Yep.
What is the microbiome? You're moving stool and that's causing weight loss? And my mind just basically exploded. And I immediately thought, has anyone ever done that in humans? And then I found the New England Journal of Medicine paper that did FMT for C. difficile.
Yeah. Yep. It's a Dutch group, yeah.
2013, Van Nood et al. And I just thought, oh my goodness.
And that's changed the way the NHS practice. So that is an impact of understanding gut microbes. It's changed the way we treat calcium C. difficile.
Huge. And I thought, this is going to be really big.
Yeah.
Why haven't I heard about this yet? I was 4 years in. I was 4 years into a degree in medicine and I didn't know what the word microbiome was. And then, of course, that has led to what will be probably lifelong fascination and a company. And we're just next week dosing the first patient in a phase 2 clinical trial, touch wood, everything goes to plan, fingers, toes crossed, everything like that. Now, going back to your question about medical schools, this has to be on the curriculum. How can it not be on the curriculum?
I mean, even if you're turning out GPs, GPs need an understanding of this in order to...
Everyone does.
Develop a diagnosis, an accurate diagnosis.
Imagine if, hypothetical situation, doctor stands up at a conference and he says, hello colleagues, check this is working, we found a new physiological system in the body. And this physiological system probably influences how you feel. It produces every neurotransmitter that we know of, or most of them. It communicates directly with your brain. It interacts with the immune system. It maybe interacts with every single drug that we prescribe orally. It produces vitamins. It, what else could it do? Now we're getting onto the microbiome's functions. Provides colonization resistance. Produces metabolites that improve the lining and the health of your intestine. Everyone would go, this person's lost the plot. But actually they're talking about the microbiome. So when you put it like that, it has to feature on the curriculum.
Yeah, it should do.
This is another physiological system of the utmost importance. Obviously I'm preaching to the converted, but
No, I think we're, So, well, I mean, it's the GMC, right, that decide on what the medical curriculum is. So
should we write a letter to them? I'll co-author.
Well, somebody needs to sort of knock on the door and say, hey, just what you've said. We need to be putting this into the curriculum. This is important, guys.
Yeah. Talk to me about colour on your plate.
Polyphenols. So the colours that you see in fruit and vegetables are due to a class of chemicals called polyphenols. And these have lots of health benefits, which is why we say, the more colour on your plate, the better it is, because you're getting more of those important chemicals. And in dark chocolate in particular, they're particularly rich in polyphenols. So if you want chocolate, I would say high cocoa content, you're going to get the benefit of chocolate, but you're also getting the benefit of the polyphenols, cocoa.
What is it about the polyphenols that drives a health benefit?
It's probably they're good for your microbiome and other things as well. So they'll get directly absorbed by the body, and there's health benefits to that. Yeah. But they're also part of the keeping your gut microbiota healthy as well, because they will be able to utilize them as well.
So they utilize them, and in return,
health benefits to you as well.
They give us some good stuff. Yeah. And we were talking about nuts as a source of food, and the concern is, I guess, quite high fat, high calorie. But there's something you said about immune system and Brazil nuts.
Yeah. So one of the nutrients that we know is good for immune system is called selenium, and Brazil nuts are one of the richest sources of selenium. So if you can eat 3 Brazil nuts a day, you're getting a good dose of selenium, which will help keep your immune system healthy.
5 pieces of fruit and veg,
3 Brazil
nuts a day, and you're sorted.
You're on your way. You're on your way.
Good. We touched on BEVs and we've touched on FMT. We have to go a bit deeper on both of those. So how important are BEVs to health, in your opinion?
Yeah, so that's still an unanswered question. I think they're important in maintaining or establishing what we've referred to earlier as microbial tolerance.
Yes.
So they're an integral part of keeping the immune system educated so that it can distinguish harmful from harmless. So it's a way of continually reminding the immune system not to react. And again, coming back to John Seaber, he was doing these experiments 25 years ago with an organism called single filamentous bacterium, SFB, which he could colonize his germ-free mice with, and showed that everybody's saying, the immune system in these animals is going to ignore them. It's immune tolerance. He actually showed that the immune system does react and recognize it, but it's a low-grade, persistent, sub-threshold activation immune system. And if you interfere with that by throwing in too much at the wrong time, it will tip the balance towards pathology, disease, and infection warning. So, it's like the conclusion of what he was doing was that you need this chronic, long-lived stimulation of the immune system just to keep it in check, to remind it that, you don't react to what's not going to hurt you.
That's interesting.
But if you mess with that through, a leaky gut and sudden influx or an invasive pathogen, too much at the wrong time, then it tips the balance. The immune system says, whoa, wait, this is bad. And that's when you get collateral damage, which we believe is sort of part of the scenario of inflammatory bowel disease.
And autoimmunity more generally.
Autoimmunity,
right?
Exactly, yeah.
Right, so the triggers, and potentially the trigger, is an imbalance between.
So the question is, how does your immune system, in what form does the immune system get these messages from the microbiome? And we believe BEVs are an important part of that.
How are you elucidating the importance of BEVs?
So we rely on a lot of animal studies to do the animal experiments. Then we've got in vitro systems where we can look at individual cell interactions. And in immune cells from all the animals we've looked at from all different parts of the body, the signature response is that BEVs elicit an anti-inflammatory immunoregulatory response from those cells. And we're talking about the sentinel cells of the immune system. So these would be called dendritic cells, macrophages, and monocytes. These are the cells that trigger the T and B cell responses, the cellular, the antibody responses. So these are the key sentinel cells of the immune system. And the BEVs instruct them to generate immunoregulatory responses. The key signature there is interleukin 10. That's the cytokine that is our signature for this type of interaction.
Also important in IBD, right?
Also important in IBD, because if you're deficient in IL-10, then you are more prone to get more severe.
So someone listening to this might know someone with IBD or have IBD, and they might be going, right, how do I get more BEVs to produce IL-10?
Yeah, so I think the first thing is to repair the barrier, because that is clearly a problem in both Crohn's disease, ulcerative colitis. You have a leaky gut. And of course, you're not going to repair in the face of a full onslaught from all the microbes in your gut. So you need to be able to allow the barrier to heal. And that means depleting the microbiome, so antibiotics, changing the diet, etc., can help with that. And once you've sealed, you then have a chance of restoring that immunoregulatory response. We've seen this in using immune cells from healthy individuals versus inflammatory bowel disease patients. They've lost that IL-10 signature. But
in IBD.
In IBD.
Yeah.
But if you can keep them in remission long enough, it comes back. So you can restore it. And that's a good indication. Then you've got immune tolerance. Your immune system's reset.
Got it.
So it's no longer autoimmune. It's now. But it can take a long time because anything that happens in that period just kicks you back into, a disease flare-up, for example.
And the BEVs are fascinating because they can cross the various different layers, can't they?
They're good at crossing barriers. So in animals, we've shown
they stay intact across the mucus layer and.
Well, that's the key thing is at some point they have to break open to release their cargo. And we're now trying to find out where that occurs. So in our tracking experiments, they can clearly get around the body as intact.
That's unbelievable, isn't it?
A lot of them, of course, will get mopped up and processed and degraded by the epithelial cells or the immune cells in the gut. But some do get across and they go to the liver, they go to the kidneys, and some of them can go to the brain. So we're just trying to identify where exactly. And are they still intact when they're reaching these organs? That's what we're looking at right now.
How do you do that in humans?
Well, in humans, it's very difficult. We would have to use some imaging modalities, using the right tag or label for the OMVs, and we'd have to get off the ethical permission to sort of feed individuals with these and then track them through MRI or some imaging modality.
And do you know, so for example, does strain X always produce BEV, Y, and Z? Or is it not as simple as that?
It's not as simple as that. These are we're talking about clever, very clever bacteria here. So they can change what they put into.
Do you think I'm really going off piece now, okay, but just allow me. Do you think bacteria are intelligent?
If we're defining intelligence based on what we believe human intelligence to be, we say no. But they're clearly adaptable and responsive to their environment, which humans are, but bacteria do it in a more simplistic way. And they change what they produce to adapt. And usually it's on the basis of survival. So they may change the types of enzymes they make in order to adapt to changing the diet, different foods. They have to produce them to break them down to get their nutrients. And so BEVs are very good sort of messengers and sensing messages. So the bacteria will package into the BEVs enzymes, hormones, things that it makes to influence other microbes in the environment, kill off competitors, or to promote the growth of microbes that are beneficial to them, or to impact on the host.
Yeah, but how does it know which BEVs to produce? I just can't get my head around it.
So, I guess the simplest way is that they have sort of sensors, and these are encoded in their genome to respond and make the genes in response to a particular nutrient or lack of a nutrient. So they have like on and off switches, which are triggered by a particular nutrient that gets into the cell.
Okay.
So depending on which switch is on or off will determine what they make or what they don't make and put into their BEVs. So, in our study, we show nutrient stress is a big factor that causes the bacteria to change what it puts in its BEVs.
And by that, you mean what you give to it?
So if we starve them or change the sugar content of the growth media, then that causes the bacteria to start producing different proteins that puts into its BEVs. Now, interesting, when you compare the BEVs that are made in a culture flash to those that are made in an animal, they're very different. So what it says, the animal, us, we in some way can influence the bacteria.
Wow.
So it gets more complicated.
That's crazy. So there's other things. So
there are other things. So we have an influence.
This brings me back to the intelligence piece. I know I understand your response, which is, are the bacteria in our gut going, why am I here? What is the meaning of my existence? Probably not.
No, survival. That's the thing.
It's all about survival.
It's all about reproduction, survival. So they will adapt to change the environment to ensure their survival. And so some bacteria will sporulate, they'll form spores because the conditions are just not right for their growth. So they'll become dormant spores.
So they just say, right,
so
see you
they'll
later.
Stay in that. And then when the right cues are available, the right nutrients,
they're back.
So
they're just so much better than us, aren't they, at survival?
So the bacteria we work with, I think, are really clever because they can, if we starve them of things, they can feed off the mucus. So they'll feed off us, basically. They switch to their metabolism.
Disappears and it becomes
And of course, if that goes on for too long, you wear away the mucus barrier and you allow things to get in, and then it's a bad case. It's a bad scenario because you're contributing to a leaky gut potential.
Bad scenario for the bugs and us.
Bad for both, because the bug, if these bugs get into the bloodstream, that's really bad.
It's a real problem.
Sepsis, septic shock. And, that's a very high fatality.
Yeah, a lot of people don't actually think about it like that, do they? It's that we are just centimetres away.
Well, hair, think of the width of a hair.
Yeah.
Width of a human hair, that is essentially what we're talking about in terms of a barrier.
We're a human hair away from sepsis.
We're a hair's breadth away from disaster.
See, it almost makes me happy because it's just so fine-tuned. And
but that's the way we've evolved.
It's just it's incredible.
It works.
Yeah, it's incredible. And it's incredible that you can be born in all different parts of the world and you still have a microbiome and you still have a mucus layer, and the mucus layer protects you from your microbiome, and your microbiome has these key functions, core functions. Yep. So what happens then if I know this is totally unethical now, and we would never do this, but there must be places on Earth that don't have many microbes, like, I don't know, desert or...
Well, they're isolating from these cores, right? They dig in the Arctic, Antarctic. They take all
where I was going with this
extreme environments.
We should talk about extreme microbiomes in space, but what if a human was born? No, they've got their mother's microbiome, don't they?
The mother is the source of your microbiome. Mums are important. They give you all the good bacteria.
Because I'm just trying to think in my head, could you ever be born in an environment where there's a very low diversity of microbes? And my question to you would be, how do they get the keystone functions if there's only a small number of bugs? But it's just never going to happen, is it?
No. I mean, you'll always...
There's nowhere on earth.
I mean, you can't get away from the human host, the mother giving birth.
Giving birth. You have to come into being.
You have to be born. And part of that process is acquiring microbes. And that's why vaginal birth is the preferred route.
Yes.
Because you're getting the benefit of your mother's healthy microbiome.
Okay. Do you think there's microbes in space?
I don't see why not.
Yeah.
I mean, it's life on another planet, right? If we look at it, life forms like us, well, not probably as us.
No.
So, there probably is. We just
single-celled organisms.
Don't know how to describe it or identify it yet.
Yeah. Carbon-based.
Yeah. I mean, going back, the Big Bang, right? One idea is there were microbes on that initial impact, that led to, the impacts of Earth with comets and meteorites and things that carried with them.
So our Earth microbiome was seeded by some other microbiome elsewhere. Yeah, I like it.
Well, it had to come from somewhere, right?
It had to come from somewhere. Why not? Why not? Wow, there might be even more crazy microbes not too far away from us.
No. Aren't there, isn't that part of the expedition to Mars is to look for these ancient...
Yes. I mean, I've been doing a lot of reading recently on lichens as a model of symbiosis. And... well, these are kind of crazy. Lichens are just a microbiome, essentially. But as part of that, I've been reading about the Extreme Microbiome projects. And what they're doing is sampling microbiomes in extreme parts of this planet. So the vents and the Arctic. And seeing if there's bugs there. And if so, what are they like? And that's important because, two reasons, really. One, if we're sending people out there to these extreme, these other planets, we need to know really kind of what we might be contending with. And the other element as well is, what are we bringing back into our ecosystems, and how damaging is that going to be for us? So that's fascinating. It's worth checking out if you're interested.
Well, I think there's viruses, isn't it? Because they can live outside of a host forever, a long, long time. So I think if it's something like that is going to come, it's probably going to be a virus.
Don't fancy that. Tell me about your work in FMT, your experience in FMT, your plans.
So we've been working on FMT for a while, and we've been looking at the context of aging. Aging. So your microbiome deteriorates with age. We know that. Even looking at...
It gets less diverse?
It gets less diverse, yeah. So, and that is coincident with physiological decline. And in particular, look at your immune system, right? It goes into senescence with old age. So we become immunosenescent, which means the immune response doesn't respond very well to vaccines. We're more susceptible to infections, and we can get predisposed to certain types of cancers because we've lost that immune surveillance, protection, robustness. So the question is, is this linked to the deterioration, change in your microbiome with aging? And of course, we've got all the cognitive deficits as well with aging. We start to lose, our eyesight fails with age. All these bodily functions fail. Yep. So a couple of years ago, we started looking at this, thinking, well, what if we took a really old mouse, so 2 years of age, which equates to around about 70, 80 years in humans, and we take a young mouse, an adolescent, teenager in human years. We took the microbiota from the young mouse, we put it into an old mouse. What effect does it have? Well, quite a lot, as it turns out.
Okay.
So what we saw was that we lost a lot of this, what's called inflammaging. So a lot of the deterioration with our bodily functions due to this chronic inflammation that happens in later life. So with the young microbiota, we were essentially able to dampen that down. And we reduced inflammation in the brain. The immune system sort of kicked up a little bit. And interestingly, the eye, we now see much more functionality in the retina.
After the FMT.
So
the FMT reversed some of the hallmarks of aging.
And of course, we did the opposite. Wow. We took an old microbiota, put into a young mouse, and we saw signs of aging, accelerated aging in the young mouse. So it's, hey,
Here we go, guys.
There we go.
We've got it.
And it's interesting
Cure for aging.
the media picked up on this, of course, and we even had a nice comment from Kim Kardashian. You know, give me a shit sandwich every day. I'm happy. It keeps me healthy
Is that what
longer.
Kim Kardashian said? Does Kim Kardashian want a FMT?
Yes,
Does she
that
want
was
the
her
best
response.
FMT available?
Somebody had talked about this. Said, "Yeah, I'm up for it. Give me, the milkshake or the sandwich, and I'll take it."
Wow. Well, soon
So
she'll be able
it's
to
had
get
an
a
effect.
capsule.
So of course, because what is in it? What is in a young microbiome that is not in an old that can have these effects? So
Can I ask one question? Sorry, the microbiology, you're talking about the whole ecology, the full
Whole.
ecosystem, not just the bacteria.
it's a stool sample
stool,
that's
Yep.
homogenized and then administered.
Were you expecting that result?
No, not really.
No.
Well, we were looking at the immune system. We thought, well, this is a good chance because I submitted a grant to the NIHR 3 years ago to do an FMT in elderly individuals to boost their immune response to influenza vaccine, the annual influenza vaccine. Okay. And they said, well, you can't do this. What's the evidence that giving an FMT to otherwise healthy individuals is not going to have adverse effect?
Yeah.
So that didn't go anywhere. So we had to sort of go back and work on our animal models.
Can I just say, if you would like to do a study on aging, I mean, my main interest outside of antibiotics is kind of aging. I'm also interested in lichens and plants and plant-based medicine.
And sushi.
I love sushi as well. I really like sushi. But I have been saying for a while, look at the diversity in these prospective longitudinal studies in patients who are older and look at how it compares to earlier in life.
Yep.
Vastly reduced diversity. I think Paul O'Toole's done a lot of work on this. At the
Key species missing as well
keystone species missing as well, right?
in the elderly.
And that could be because their diets change. It could be because they're in a care home. It could be because of concomitant medication. It could be because they've got underlying disease. But if we just assume for 1 second that it might be to do with aging, why don't we do an interventional study in humans? Are we expecting to see a negative impact of increasing microbiome diversity?
Well, I think it's because the attitudes are hopefully have shifted and changed since I submitted that grant.
Yeah.
And, we've done the experiments in mice. We're going to do a similar experiment in a primate model. On the back of that, and we've got an FMT lined up for ME/CFS, which have...
Definitely need to talk about that. Yep.
So with that, I'm thinking now the NIHR cannot say, you haven't done enough work in animal models, preclinical models, or human, because we will have done by then. And I'm going to go back in and say, look, with the pandemic, influenza epidemic, potentially a pandemic, why wouldn't you want to protect the vulnerable with a simple procedure? And by the time we get to that, I'm hoping we'll have identified what the active ingredients and the missing bugs are, so we can just give a defined mixture
Of bacteria.
of bacteria. Well, maybe not, because in our mouse model, we've shown some beneficial effects in just transferring fecal water rather than the whole stool. And of course, fecal water contains viruses, BEVs, metabolites, other nutrients. So clearly, maybe you don't need the bugs. You just need what they produce.
Should we talk about that then? Because some people talk about this sterile fecal filtrate. Sterile means no bacteria, right?
Yeah, but it doesn't mean there aren't viruses, which
There's
are not
definitely
sterile.
viruses, right?
Yeah.
There's definitely viruses.
Yeah.
Because there was a study in C. diff where they gave this, what they called sterile fecal filtrate. They did a pressure filtration and they gave it to like 3 or 5 patients and they had a positive result.
Yeah.
And the hypothesis was actually, do you need the bacteria at all? Or do you just need their products of metabolism? But
So if that's interpreted as an effect due to metabolites, that's going to have a short-lived effect when you transfer them. So that's the benefit of giving this whole stool
is
you've got the producers there.
Yeah, you've got the factories.
Yeah, it persists.
Yeah.
Viruses, again, you say, well, eventually they'll decline because if
They'll
they're
disappear.
They'll disappear. So I guess it depends. You can pick a scenario and a treatment for a particular case. And you're right, maybe you don't need a whole stool. You can give the sterile filtrate.
Yeah.
Or a virus cocktail, or BEVs, or metabolites.
Yeah, and maybe it depends on the indication.
Yeah.
Can I just ask you about the ME work then? And also, what's your vision for FMT and longevity? Do you think we might be I mean, Kim Kardashian to one side. Because people have, for a while now, done these, they've tried to have blood transfusions from younger people. There's companies that have tried to do this, right? You've probably heard of it. And if you've got enough money, you can kind of, in this day and age, try anything.
Yeah, sure.
So I'm surprised that there's not more of a demand for...
But let me put it this way. Don't you think it makes perfect sense? Because we've been talking now about the microbiome being essential for health. If that gets messed up, maybe it's not surprising we are going to get sick. And aging, if you look at it context of a disease, then the implication of that is treatable. And where would you want to start the treatment?
Aging's a disease. Yeah.
Is it? Is it a disease?
I think it could be a disease, yeah.
So
I think it could
the
be
microbiome essential for this. We know it deteriorates in parallel with aging. So, if we change the microbiome, could we at least slow the progression? Benefit is we stop it. But anyway, we can slow that trajectory. And that has a big impact on whether or not you develop neurodegenerative diseases in old age. We haven't talked about Parkinson, Alzheimer's, which we're interested in as well. So, you could put the brakes on not just declining health in old age, keeping people fitter and more active for longer, could have an impact on these really severe debilitating dementias. And, my work on ME is related because they have significant cognitive deficits, GI issues.
For the listener, fatigue. Can we just provide a little bit of background on? So
we're talking about myalgic encephalomyelitis. The common name is chronic fatigue syndrome, for which there's no cure, there's no effective treatment, and the number of patients is really underestimated because it can take patients years to get a diagnosis. And there is, I think, a bit of a ticking time bomb here because with long COVID, I suspect a significant proportion of those patients will develop ME. So the numbers of affected patients are only going to get better. And so few of them make a full recovery. And the more severe forms, patients are bedbound, housebound, incredible sensitivity to light, sound. So these people are in desperate need of new treatments. And we're fortunate in working with a charity called Invest in ME Research, which is funding research at the Quadram to develop a centre of excellence for ME. They've supported with the university PhD studentships. They're funding an FMT trial in ME, which will hopefully happen later this year. So we're looking at trying new interventions, but not trying to uncover what the root cause of ME are and develop new interventions to treat ME.
Your thinking is that if you instill a healthy ecosystem, you drive processes in a different direction to what might be contributing to the disease?
Yeah, so the disease has long association with virus infections, and it was first thought to be a variant of polio.
Really?
Infection.
That's the history. That's fascinating.
It goes all the way back to sort of, this is just another form of polio.
But also it gives you some indication as to how terrible this is.
Absolutely, yeah.
If that's what in the early days people were thinking, right? That's how bad it is. Yeah. So for the really severe cases, can patients actually leave home?
No, they can't even leave their bed.
Wow.
Yeah. And it's a darkened room. They're sensitive to light and sound. So just the touching their skin. I mean, we've seen some of these patients and we're trying to recruit them to some of our studies. And, it's just a horrible, horrible existence.
That's just really, really bad. And there's nothing available.
No. So they were originally described in outbreaks in places like Iceland, America, in the UK. But the exact trigger's not been identified. There have been links to herpes virus, Epstein-Barr virus, polio virus, as I said. Yeah. So we believe that viruses are probably a trigger, but in fact they're not acquired. They could be from what's normally part of your virome that makes up your gut microbiome. Some patients, they get a bloom or an outbreak of these viruses, which can trigger symptoms of ME. And if it's not treated, the virus is eliminated. It's a chronic exposure. So we believe that treating the microbiome might be an effective means of treating ME. And FMT is probably the more radical approach.
Right.
It will replace what's there with a healthy microbiome. Yes. So the FMT trial we're proposing to do is important. And it's based on case studies carried out in Australia where particular individual's been using FMT to treat his patients very successfully, apparently. So we're going to want to take a clinical trial, placebo-controlled,
gold standard,
randomized, the gold standard.
Yep.
And this will say, FMT could be safe and effective treatment for ME, which would be huge
Transformational
for patients.
Transformational for patients.
Huge.
And have you found that there's been already quite significant patient demand?
Yes. So
they've probably, my experience, I mean, you're describing something which is just, I can't even imagine. I can't actually imagine it because I'm so active and love being outside. But, you know, in my experience, patients do a lot of research. So they probably have read the case reports that you're talking about and want to find an FMT.
Yes.
Right.
So I gave an interview on BBC4 Radio Today programme 2 weeks ago, and I must have had 70, 80 emails from patients desperate to sign up.
I believe it. I believe it.
And they're still coming in. I looked at my emails today, and I've got half a dozen patients wanting to know more about signing up for the trial.
We need to help these people.
Absolutely.
We need to help these people.
So we're fortunate in the part of the country where we are that there is the chronic fatigue centre based in Lowestoft, where they have they treat a large number of patients. So they will be our recruiting centre.
Yeah.
And they have over 1,000 patients registered with them, and they already have over 200 patients on the waiting list for the trial. So I don't think recruitment is going to be an issue for us. There are patients desperate.
Yeah, I believe it.
Desperate.
Well, all the very best with all of that.
Thank you.
I'm really excited, actually, to see what happens. I've been talking for a while, but I would love to talk about Parkinson's and Alzheimer's if we can.
Sure.
And what the links may or may not be there and what the future might be for these patients. And then let's talk about the future more generally.
Okay.
And then I'm going to ask you about probiotics and prebiotics. You must all the time get people saying, Prof, which probiotics should I take?
And my answer is don't waste your money. Eat more fruit and vegetables.
Yeah.
Feed your gut microbiota.
Yes.
Because, what you consume in a little yogurt pot is literally a drop in the ocean. Which is fine if you're selling it, because then you have to eat every day to get that. But far more effective is to feed your gut microbiota. Give them what keeps them strong, healthy, and you healthy. So spend your money on fruit and vegetables.
Get your 5 a day.
Get your 5 a day.
Don't spend £60, £70, whatever it is.
No.
Spend it on the best food you can afford.
Spend it on the best.
I'm really pleased, Professor, because that's what I say to people as well. So I'm very happy. I'm singing from the same hymn sheet as you.
Good.
Yeah, you are what you eat, right?
Exactly.
Yeah, that's it. Good. So now to Parkinson's and Alzheimer's. So what's the thinking there? Everybody knows somebody with.
It's interesting, and I think the microbiome is sort of coming a bit more front and center in terms of a possible causal link here. So with Parkinson's disease, again, a lot of patients report GI issues prior to onset of symptomology. Dopamine, the key hormone, the neurotransmitter.
Yep.
Over 50 to 60% of that is made in the gut. So, if you have a dopamine deficiency in your gut, that's probably going to impact on the overall balance.
It's huge.
And that could be important as well. and there are sort of studies built in animal models which seem to link severity and onset to the types of microbes. And you can alleviate some of that by manipulating the microbiota.
Right.
So there is a keen interest in this. And I think studies in patients have shown their microbiota is clearly different to a normal healthy individual. So there is, I think, reasonable grounds on which to say, okay, if we can intervene more wholesale early, so pick up the first symptoms and then give a wholesale replacement of their microbiota, we might have a better chance of just sort of kicking it around the edges with probiotics, antibiotics, etc. So FMT might be beneficial in those individuals. Alzheimer's disease, again, a lot of the evidence is based on animal models. But it's interesting because there's evidence that some bacteria can produce things that mimic the fibers that trigger some of the pathology seen in Alzheimer's patients. And there's some believe that if you've got these bacteria in your gut producing these fibers, they can get into the vagus nerve, get into the brain. This then seeds the development of these plaque forming that we have in the gut. So that's one line of thinking. The other, of course, it's due to what the other things that the gut microbes are producing, and it's tipping the balance in neurotransmitters, et cetera, et cetera. So there could be a direct causal link there through microbes producing things that mimic some of these pathogenic proteins that are found in the brains. And this then just exacerbates the inflammation and progresses the disease. Wow. So again, the argument there is, if we wholesale replace what's there with a healthy microbiome, maybe we can stop that, slow it down.
Yep.
So the Quadram, on the back of a trial that we want to do, an FMT trial, quite soon, is in Parkinson's, and particularly early onset, and working with the hospital, the Norwich University Hospital, there are consultants there who have identified what could be prospective candidates for the trial. Early onset, relatively young people, so if there is a beneficial effect, have a major impact on the rest of their lives. So that's what's sort of in the pipeline, as it were.
That's very exciting. Very, very exciting. I think everyone listening will know somebody who's been affected or, have had first-hand experience of how horrific they are. These two neurodegenerative diseases.
Absolutely, yeah.
It's devastating. So what does the future look like then for this field, in your opinion? And if we had this conversation again in 10 years, 20 years?
Yeah. So I think the future is prevention.
Yeah.
So we're getting very good at cataloguing the microbiome and clearly, to increasing powers of resolution. So what I would hope is that we're moving towards personalized medicine in the future. And then when you go to see your GP, you'll take a stool sample. The GP will have the equipment, tests, whatever, to be able to immediately analyze your stool sample and say, missing this, missing that. Ooh, too much of that. This is bad because this could potentially put you at risk for developing cancer, IBD, rheumatoid arthritis.
Right.
So we need to do something now about that. So depending on what's missing or what's being overproduced, the doctor will be able to come up with, this is what I'm prescribing. It could be a defined cocktail, capsules. It could be changing your diet. It could be phage therapy, okay? So the beauty of using bacteriophages is they're exquisite in terms of what they target. They target just specific bacteria that have the receptors that allow them to bind and then kill the bacteria. So antibiotics are a shotgun, phage are like a sniper rifle. They can home in on the bad guys and leave everything untouched.
What level of specificity? Strain level?
Strain level.
Perfect.
So, it could be a phage. We're going to give you these phage cocktail that you take. We're going to knock out the bad guys. We're going to change your diet to boost the levels of the good guys. And, we'll give you these BEVs because they've got all these nice things in there that will trigger the right sort of hormone release in your body. Or we'll give you this cocktail of bugs which you're missing, supplement. So the GP will have an array, an armory of tools to be able to decide what's going to work for you.
Yep.
Personalized medicine.
And everyone's going to have their 5 a day.
And everyone. So the exactly. So the important thing with all of these treatments is you've got to enhance, you've got to promote them, you've got to keep them alive, you've got to keep them going. So that comes into your diet and your lifestyle. Okay?
Yes.
You've got to live healthy as well as eat healthy.
So what do we do in our FMT studies then? Do we give, I say we, I'm asking you, but I'm also asking myself, are we asking them to eat a particular diet after?
Well, we do. I mean, the hospital does. When the patients come in for C. difficile treatment by FMT, they're given some advice about what to eat. Yeah, post-treatment.
That makes a lot of sense.
And obviously, it's things to avoid. And then you try and get them to eat more healthily. So you try and sort of eliminate the things that will knock out your beneficial microbes. And, we had a bad incident in one case. It was an alcoholic who came in for a recalcitrant C. difficile, was given an FMT, and they said, look, you're going to have to stop consuming so much alcohol. This could impact on your FMT.
Yeah.
Unfortunately, they couldn't do it, and the FMT was useless.
Didn't work.
Didn't work in that individual.
Because they were drinking so much?
Because they had gone back to the lifestyle that probably contributed to the infection in the first place.
Did we give them another FMT or?
Well, that's the moral dilemma, isn't it?
Do you keep doing it? Yeah.
Do you keep doing it if they're not going to change their lifestyle?
Didn't change their cravings.
Yeah. I mean, it's the same argument with smoking.
Yeah.
Right.
Yeah. No, I hear you.
You know, it's a personal choice.
Yeah.
But society bears the brunt of that choice.
Absolutely. No, 100%.
Yeah.
Professor, we're at 2 hours, believe it or not. So
3 hours.
2 hours.
2 hours. Okay.
Yeah. Time flies when you're having fun.
It does indeed.
So, just wanted to thank you for coming on.
Well, thank you, James. It's been a pleasure.
It really has been a pleasure. It's super, super insightful and interesting couple of hours, and wish you all the best with your research, and look forward to staying in touch and maybe collaborating.
That would be great. That'd be fantastic.
Thank you.
Thank you very much, James.
Cheers.